It reminds me, I used to, when we'd have free periods at school, I would take those TVs that they wheel in for educational lessons
Yeah.
that weren't actually connected to a cable, they just had a VCR. But if you put like your earphone jack into the back of them and you wiggle them at just the right angle, you could use them as antenna. That's what I feel like this
Really?
recording is like.
That's insane. I never knew that.
It was, yeah, it was a fun hack that we discovered out of desperation.
There you go.
That sounds like fun.
All right, I am very, very, very excited. Firstly, to welcome all of our listeners to another episode of Science Actually Presents the Nerd and the Scientist. But the reason I'm excited is because this week we have a very special guest, Zoe Xirocostas. Am I pronouncing your surname correctly?
You got that perfectly.
Okay. Whew. I was worried. I was messaging Ina this morning and I said, I learnt so much Greek during undergrad, but I need to see the letters in front of me. So Zoe, you're a Chancellor's Research Fellow at the School of Life Sciences at UTS, the University of Technology, Sydney.
That's correct.
I'm just kind of like in awe, like the publication record that you have, the stuff you do in science communication, the fact that you are leading a research lab, incredible.
I'm so flattered to hear you say that. Because I do suffer with imposter syndrome like a lot of people in science do as well.
How?
I don't know.
Because you're not an astronaut also? Is that the missing piece or something?
I think I'd be too scared to go into space, if I'm honest with you. But
Really?
well, I would love to go into space, but I think I'd need to be sedated on the way there, I think. Yeah,
Like
on the way
Diego
there.
Rock is from the A-Team.
I mean,
Imposter syndrome is very real, though.
Yeah, and I mean, I've spoken to professors and been like, because it comes in waves, right? It's not all the time that it happens. And I've spoken to professors and I'm like, when did it stop for you? And they looked at me and said, Zoe, it never goes away. I'm like, oh great. That's not what I wanted to hear. But the good thing that I also was told about it was it means that you care about what you do and you love what you do. So you want to know that you're good at it. So it's a double-edged sword, I guess.
True. I guess it keeps you honest in certain ways. I guess my question, given that, so like I was, obviously doing a bit of like LinkedIn stalking to try to like background and understand who is this person with this illustrious career at such a relatively young age. And my thought was, surely you have to have like the highest level of self-confidence to be like putting in for grants and putting in for these positions.
Or delusion.
That's awesome. There's a level of insanity that goes with it.
You know what it is? You do have to have confidence, and I'm like, I do have confidence in myself, but then again, like I'm applying for another grant right now, and this whole time that I'm writing it, and I'm like, oh my God, I'm not good enough for this. But you've just got to kind of persist and hope for the best, and just have a team of people around you that you can lean on and that supports you and that encourage you. Because I don't know, I guess from the outside, people might think, oh, she's very confident, she'll put in for grants and things like that. But it really is encouragement from others that does help drive me as well. And also, wanting to stay in the field of science, putting in grants does help with that. And, I've taken the career path of sort of applying for fellowships, and that allows me to create my own sort of research journey and what I want to study. And I don't really have to rely on postdocs where I'm under sort of under, the creative direction of the research that my supervisor wants. I can really drive my own research direction if I write my own fellowship. So yeah, I don't know. Maybe I'm just too, I don't know, too stubborn and just want to do what I want to do. And so I'm like, well, I have to write a fellowship because I just want to do what I want to do, and hopefully it works out. So yeah.
I think stubbornness is the key, and I really relate to that. And also, I was listening recently to the wonderful podcast episode you did on convos with chordates
Oh yes.
with Sammy and Anastasia.
Yeah,
For folks who aren't familiar with it, I highly recommend you go and listen to it. The
they're
hosts are
great.
wonderful, and
Yeah.
the guests are even better, um, if I can say that. But like this episode, you were all discussing both like the different non-traditional aspects to your career paths, and I guess the stubbornness that was necessary, right, to keep going once more into the fray, right, to like keep going at it like every time you get
Yeah.
knocked back. And I feel like there's so much of academia that is not about being like some hyper brilliant lone genius who doesn't need help from anybody. It's I'm a hard worker. I've got a brain in my head. I'm just going to keep going at it.
I
Yeah.
completely agree with all of that. Honestly, I don't see myself as the smartest person. I don't think I'm super intelligent, like wow, Einstein level person. I just think my stubbornness to finish the things that I start and that determination is what has led me to like continue on this career path. And you need to it's unfortunate, but you need to fail to progress, I think,
Yes.
at least.
Yes.
So so people always see the highlights of your career, right? They say, wow, she got this fellowship, she's published all these things. You didn't see the awful, awful peer reviews I've gotten on some of my papers. Absolutely awful. Like I read it and I had to like take the rest of the day off. And some of these things that were said were like, you know, I don't trust this science, blah, 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 blah. Yeah, just like really, really like
Wow.
targeted horrible things that like my supervisor at the time, I just finished my PhD at the time. My supervisor was like, wow, I'm not sure how the editor let that go through to you. But do you know what? I published that paper in a different journal 6 months later. So like it really wasn't, yeah, just one person's opinion of a study that I did really didn't matter in the grand scheme of things. It eventually got published. Like I worked with a team of 16 people. I thought surely 16 scientists couldn't have created something so bad that the whole premise of the work of my PhD is completely turned to crap. Like surely not. Yeah.
It's
And
because
then
a lot of the work you do is to do with the study of climate, and that is the science of studying it, but it's also a very politically charged topic, and
Ooh.
that's probably where some of the pushback came from.
well, so that particular study was actually looking at introduced plant species and the mechanisms that facilitate their invasion.
Got it.
So that wasn't it, but I can
Wow.
definitely see with my future work that playing a part of it potentially.
Yeah.
Yeah.
How do you
Look, you'd
plan?
hope not, because most scientists agree climate change is happening. There is so much data out there. We're
Yeah.
seeing the effects of climate change, of how it's changing the phenology, the timing of plant species flowering,
Yeah.
of the emergence of pollinators. Like with the classic example of the cherry blossoms in Japan, which we have
Yeah.
data from. I think it's the last 400 years. I would say we have data, maybe even more, and you can see how the timing of the blossoms has gone from like mid-April to towards March now, which is really worrying. Yeah. Oh, but one thing I did want to add in that before I went on my tangent, which I tend to do,
Lo-
sorry. So
Love a
No,
good
please,
no.
tangent.
please pull me up whenever, if I'm going off into my own world. But what I wanted to say was that before I got the fellowship that I have now, I actually applied for one a year prior. I got through the expression of interest stage. I wrote up the whole fellowship. I got invited for an interview, and then I didn't get it. And yeah, and that was an 8-month process. That was 8 months.
For context, Benjamin, there's like a lot of these grants in Australia. So currently, there's a big one that a lot of my friends and colleagues are applying for. That takes like 2 to 3 months of work to prepare your initial application, of which I think like more than half of the first round, like the EOI stage for DECRAs get rejected.
I'm not sure. Also, there's some construction happening, and I'm really sorry if you can hear that.
Oh, we can't actually.
Okay, good,
Cannot.
good. All right, good. Sorry. Yeah, so with DECRAs, I'm actually applying for one right now, so I'm in the thick of it as
Yes.
well. Are you applying for one, Kovi?
I'm not. No, I'm just
Okay.
doing regular old postdoc applications at the moment.
Yeah. Okay. Sorry, I didn't mean to jump on
No, no,
parents
no.
thinking at all. That was not me. That was just me saying how stubborn I am with my own research sort of ideas that I just don't want to let go of them earlier.
It's good. That stubbornness, we like it.
Yeah. But yeah, so for a DECRA, you have to express your interest to a university around Australia and say, this is the project idea that I have. You'd probably give them your CV as well, because it's really intensive for the research office at whatever administrating university that you're at. It's really intensive for them to provide that support to you, because they will do compliance checks on your grant, like all these things that just seem so over the top. But compliance checks, making sure that, everything complies to the Australian Research Council's guidelines. Strategic reviews, like someone at the office will read through your grant and look at the strategic goals of the Australian Research Council or of the Australian government and say, okay, does this line up with this strategic goal, blah, 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 or maybe you could strengthen it here. They put so much time and effort into it. So yeah, so then only a select number of people, I'm not sure how many, to be honest with you, maybe half of the people that apply for an expression of interest or a notification of intent to submit, they call it notice, NOITs. I don't know. They have all these acronyms.
I hate acronyms.
Yeah. So once you find out that you've made it through to actually apply for this big grant, then it's full steam ahead. There's so many different components to it. And yeah, basically everything else that I'm doing at the moment has been put on hold so that I can finish writing this grant.
It's wild. Like, there are so many other people in the same boat, and most of them, like, thoughts and prayers with your application. I really, really do hope you get it. Like, looking at your CV, you do deserve it.
Yes.
But there are so many people who are spending time, and most of them won't get it, and it just feels like such a bad system that you end up with people wasting like 2 to 3 months of what would otherwise be good research time, right?
Absolutely. And I mean, I've got 3 glasshouse experiments that I'm gonna start this year, but I've put them off obviously to finish writing this DECRA, and then I'll start doing my experiments. And I've got all these other collaborations that I'm part of that I've had to push back for this. So yeah, it does take a lot of time for sure. And I'm so aware that the success rate is very low. I think last year the success rate of getting one of these was 8%. Because the DECRA, so it stands for Discovery Early Career Research Award, and it's open to anyone in Australia or overseas who will perform their research in Australia. And basically, anyone in any field can apply. So if you're in law, engineering, science, whatever, you can apply for one of these things. So each year, apart from this year, there is a funding cap of 200 DECRAs being awarded. And there's probably I don't know how many there are, but I'm guessing maybe 50 applicants just from my university.
Wow.
That I'm just guessing. I don't actually know, but
It seems like a reasonable guess. Yeah.
yeah, and I know some people within my specific field of life sciences who are applying for it, who made it through that expression of interest round. And I'll be completely candid. I'm not only applying for a DECRA because, yes, it will allow me to continue on my own research path, but it will guarantee me a job for another 3 years. The sad reality of the state of research and science in Australia is that we are relying on these young early career researchers and PhD students to do the majority of research. And you've probably already spoken about this on your podcast, but we're relying on all these young researchers to do the majority of the research and a lot of the times formulate these ideas as well. And then we don't support them into ongoing, permanent, stable roles.
Yeah.
And then you lose a lot of that talent because people will think, okay, if they can, they will say, okay, well, I'll just go overseas and go to Europe or somewhere because, and I'm not saying Europe is like the fix all to Australian researchers' problems, but there is a lot more money invested in science in Europe. So there tend to be a lot more research opportunities over there. It's really hard to find.
It's a whole ecosystem. It's a research ecosystem where like we see the impact on the how difficult it suddenly is, even more so than it previously was, to get research funding or a post PhD research role in Australia because of what's happening in the US, for example.
Yeah.
And then I have a friend at a Canadian university recently who's telling me that a role that in the past they've had 50 to 100 applicants for, they had 450.
Yeah.
Wow.
For one role. Like it's tough, and I think that we need to do more as a society to maybe realign our goals, and or even if there are things like that we say are our goals. So for example, climate change, right? We have a consensus, and yet I don't think the research funding on climate change related research necessarily reflects that. So I think that there's definitely some soul searching that we could be doing as a global culture, if not at the very least as a national culture here in Australia.
Yeah, absolutely. And I mean, it just, I just don't see how we're allowing this to slide because the university sector and the people that make all these decisions, they're smart people. They're not silly. Well, you're rolling your eyes there. But surely they understand how continuity works and that if you're constantly dropping people or people are leaving the research landscape because they know that it's not a viable sort of stable career path, because you've got to think a lot of people at this stage of their lives, they're also having families, wanting to buy a house, things like that. How are you going to do that if you're on a contract to contract every year? What bank will give you a mortgage, or how stable is that going to make you feel to bring a child into the world where you're thinking, oh, well, I don't know if I can have a baby now because by the time I get pregnant and have the baby, I might be out of my contract and I won't get parental leave.
Ugh.
Yeah. And I come from a Australian privileged scenario where we get parental leave, because I know in the US that's not always the case. Correct me if I'm
We
wrong.
talk about it a lot.
Yeah.
Checks out.
Yeah.
No, we have parental leave in some states. State differs.
States. So it's not nationwide.
There's nationwide, but then the states to states like California, where I am, it's way more than other states like Arkansas or something.
Yeah, wow.
I don't know how all the different states are, but if you just pick a random state in the South, that's generally the idea.
Jeez.
No offense to Arkansas, by the way. You guys are fine people.
Damn it! Now we're never going to get a guest from Arkansas. Thanks.
You've got to make that your goal now.
So let's say everything goes forward as planned. You
Yeah.
get the DECRA thing. What do you have? What do you hope to have to do? What are you hoping to do after that happens?
Well,
What's
look,
the
even
big
if I
first step?
don't get the DECRA, I'm still on my current fellowship, and I'm almost halfway through it. So I've still got another 2 years left. So, it's not total doom and gloom and turmoil.
Sure.
Even if I don't get the DECRA and when I finish, it's still not doom and gloom and turmoil. I have to always tell myself, you're not going to drop dead as soon as your job finishes. Like, it's fine. But so what I'm planning to do in my fellowship, and for some backstory, is my PhD was looking at mechanisms that facilitate invasive plants in Australia. So what I did was I went around Europe for 4 months and looked at the antagonistic interactions that plants had in their native range. So I looked at how much herbivory they were receiving, so how much they were being eaten by insects and things. And then I compared that to how much they were eaten by insects in Australia. And I found that was a lot lower in their introduced range. So that's sort of, and we call this mechanism enemy release. So when a plant or an animal moves from its native range to a new area, it sort of leaves behind all of those co-evolved enemies that would usually target it. And you can think of a classic example in Australia as well is of the cane toad. So when it moved from South America, when humans introduced it from South America to Australia. And fun fact, the cane toad was actually introduced to Australia to act as a biocontrol for invasive cane beetles that were eating all the sugar cane in Queensland. So these cane beetles were eating sugarcane in Queensland. So they were effectively released from their enemies. So the Australian government said, okay, let's bring in their natural enemies as a biocontrol, the cane toad. And so they brought the cane toad over. The cane toad obviously has all these novel new weapons that Australian predators weren't accustomed to, which is their toxins, the bufotoxins. And that just meant anything that tried to control these population of cane toads would just die because they can't process those bufotoxins, unlike what their co-evolved, predators in South America would have been able to.
Right.
And also, the cane toad, may I just add, wasn't even an effective control of these cane beetles because cane toads can't jump.
Oh.
They can't. They can jump sideways to move across, but they can't jump upwards vertically. And so they couldn't actually reach the cane beetles on the sugar cane. So it was just a bit of a failure on all regards, unfortunately. And now here's a twist, because of climate change, latitudes further south of the equator are warming up. So before, cane toads were only accustomed to those really tropical areas of Queensland, but now we're seeing them start to migrate further and further south. And we've got cane toads in New South Wales now,
Ooh.
which is more temperate climate, definitely not tropical, at least most of the year.
Most of the time, yeah.
Yeah. So that's my backstory. I looked at invasive species. I also looked at mutualistic interactions, so pollination, and compared pollination in plants in their native and introduced ranges. And I also built my own app. Well, I had a developer help me build an app. But we tested it, and it was to improve researchers' ability to estimate herbivory. So if you look at a leaf and it's got holes in it, the app would basically help train you on how to accurately visually quantify the amount of herbivory that was on the leaf. So like you'd look at it, and if there's a little hole, you'd be like, oh, maybe that's 1% herbivory. And then the app would tell you, no, it's actually 3% or whatever. And it'd give you feedback on how well you've done at estimating herbivory on a plant.
And
Does that
in
make
this
sense?
context, you're talking about like the amount of insects or herbivores that are eating that plant.
Just the amount of damage that the plant or the leaf has got. So like if you look at an image of a leaf
Like a
and there's
surface area thing.
yeah, like a flat scan of a leaf and there's
Mhm.
holes in it, just saying how much of this leaf is
Got it.
damaged, has been eaten.
Okay.
Yeah. And so that was born out of the fact that when I went over to Europe to do these sort of sampling expeditions, there wasn't a good way. So other studies have been like, yeah, we visually estimated how much damage each leaf had, but there was no standardized protocol or way to do that.
Right. Subjective.
Yeah, it was very subjective. So and also a lot of studies have said that people tend to overestimate how much damage there is when you're visually looking at it, because there's apps where you can um, take a photo of a leaf and it will tell you how much of the surface area is damaged or has been removed, but that takes a long time. So you have to actually take a photo of the leaf and then upload it to a computer, and then run all these different sort of analyses to do that. And yeah, it just takes time. Whereas if you're out collecting data on 11,000 leaves that I did, yeah, you can't do it by scanning an app, like doing it with scanning every single leaf, into putting it onto your computer, processing it. It just takes way too long. So yeah, I did that as well.
I'm still kind of like, I think Benjamin's muted.
Oh,
What's that?
I was muted. Yes, I was asking 11,000 leaves or 11,000 plants.
11,000 leaves.
Oh wow.
Yeah.
Still, that's a lot. Either way,
But yeah,
you're close.
I did that on how many? I did 10 leaves per plant. So that's 1,100 plants.
Yep.
Yeah.
100.
Wow.
Quick maths. Yeah.
I'm still like picturing you gallivanting around Europe for 4 months, just taking selfies with plants, just like.
I did some of that. I went to some pretty amazing places. Yeah.
It's so cool. And so I guess, yeah, to Benjamin's point, so if you do get the DECRA, or like looking forward,
Yeah.
research wise, like if any funders happen to be listening to this
Yeah, yeah.
particular episode,
Please.
what's the next step? Like,
Yeah,
that
the
you're-
next step. So the next step, which is what I'm doing now, is looking at how plants' capacity to adapt to climate change might be influenced by the habitats that they are in. So I'll run you through my thinking of this and of
All right.
my idea.
I'm
So
excited.
basically, you can think of two habitats. You can think of a really complex sort of rainforest type of habitat that has a lot of trees. You maybe have some shrubs, you have understory, you might have vines all over. It's really complex. As you walk through it, it might be, you might, have a patch of light opening up, and then, two steps later, there's a really humid pocket and it's a little bit of a ditch. And just the microclimate diversity is a lot more heterogeneous in a really complex habitat that's got a lot of species diversity. And then you go on the other side of the spectrum where you have a really homogeneous habitat. You think of a really homogeneous habitat like a grassland, where it's just a field of grass. There's not really, different canopies, different structures. So there's not really going to be much variation in sort of microclimate or the different sorts of habitats that the organism within these ecosystems is going to encounter. Okay? So if you're a plant, say it's the same plant species, say you're a dandelion, to keep it really simple,
Okay.
in a field like a meadow versus a rainforest, your offspring need to be able to adapt to wherever they might be dispersed within that ecosystem. If you're in a really complex ecosystem, you will need to have the ability to adapt to increased light, increased temperature, maybe decreased temperature in terms of the mother plant, what the mother plant was conditioned to. Whereas if you were a dandelion dispersing your seeds in a grassland field, for example, the conditions that those seeds will encounter are not likely to be that different to the mother plant in that grassland, right? So it's this idea that the complexity of a habitat might moderate the capacity for a plant to adapt to climate change. Now, the way I'm going to test this, or the way that I am testing this, is by taking seeds from plants from different ecosystem types with varying complexity and growing them in a glass house under ambient and elevated climate change conditions. So that would either mean increasing temperature, reducing precipitation, and potentially, if I can get our climatron chambers to work, increasing the carbon dioxide as well. And the way that
Wow.
you measure how much of a capacity that a plant has to adapt to these future conditions is through something called phenotypic plasticity. So phenotypic plasticity is pretty much similar to adaptive capacity in the sense that it's the ability for a plant or an animal or any organism to moderate its morphology or how it looks or how it behaves or how it physiologically sort of operates depending on its new conditions. So the difference in the traits between these elevated temperatures versus ambient temperatures, the sort of the magnitude of difference between the offspring that originate in the complex habitat versus the less complex habitat, I'm sort of suggesting that there's going to be a much greater sort of plasticity in traits in the plants that are from those more complex habitats.
This is kind of reminding me, and maybe this is a stupid comment, but it's reminding me of studies that I heard about with epigenetics of C. elegans worms, right? Where
Yeah,
they would test to see, like, if you take a generation of worms
yeah,
and you put them under extreme circumstances in terms
yeah,
of like less food,
yeah.
less water, whatever, are their offspring more hardy and can they live in similar conditions?
Yes, that's exactly it. So that's pretty much bang on. Asking whether yeah, no, you've just translated it from another sort of field. So that's great. Basically asking whether we can predict whether plants that are from these more complex habitats where they might experience greater variability in their conditions, if they're better able to adapt to future climate change conditions.
Very
And
cool.
then further along to that, if we can say that that's the case, then that's also really strong motivation for us to make sure we don't reduce biodiversity, because having biodiversity is what allows us to have these complex habitats. If biodiversity, starts to get lost, if plant species are becoming extinct, that complexity in the ecosystem reduces, and therefore, if my theory is correct, the ability for these plants or animals to adapt to new sort of environmental conditions, i.e., climate change, will be reduced.
When you're doing all these tests on plants and changing their environments and the the air and the soil and the light and everything like that, are you inadvertently creating some sort of new super plant or new species of plant? Like you have a plant that was used to this type of condition, and now you have a similar looking plant that is more better suited now to a harsher condition.
That
Is that a
is
new animal or a new
that
plant?
Benjamin, that is such a great question, because this is something that is sort of being talked about how our future ecosystems, because we know climate change is already impacting our ecosystems and they're not looking like what they once were, at least some of them aren't. Are those new conditions going to effectively turn those species into something else? And we call that rapid evolution. So rapid evolution, my PhD supervisor, years back, and I don't know if she's published the actual new taxonomic description yet, but what she found, and what one of her PhD students, Claire Brandenberger, who works at the Royal Botanic Gardens in Sydney now, um, she found that a daisy, a beach daisy in South Africa, native to South Africa, which ended up in Australia, I think in the 1920s, or maybe a bit later. So it's only been here for about 100 years. They found that due to this daisy being in a new environment, it's evolved and adapted so much so that when you grow seeds, so in the same glass house, when you collect seeds from this daisy in South Africa and you collect seeds from this daisy in Australia and you grow them under the exact same conditions in a glass house, which was done at UNSW by Claire, these plants looked different. So the height of the plant was different, the shape of the leaves were different. The flowers in the Australian populations of this daisy species, the flowers were much more reduced in size. And then, but the ultimate test is, right, okay, you can look different, right? Like, look at dogs. You have Great Danes and Chihuahuas, they're the same species. The real test of knowing if something is a different species is if it can reproduce, and if it can reproduce and produce successful, viable offspring. So what they did was Claire and Angela basically took pollen from the South African and the Australian populations and cross-pollinated them. And the seed viability was very, very low. So low, in fact, that it was basically declared that if these two populations were growing in the wild, they would be reproductively isolated. Even if they were growing right next to each other, geographically they're not isolated, but reproductively they're isolated because they can't produce viable seeds by cross-pollination. So therefore, this has to be a new species. And that's within 100 years of this daisy coming from South Africa to the east coast of Australia.
Wow, that's crazy.
Yeah. So when you say, could climate change cause new species to occur? I say we've seen it with this beach daisy, so why couldn't it happen with any other species that might become geographically isolated or become very different to what it once was?
It's ridiculous.
Yeah.
It's wild.
But also, some people might also say, but that could just be regular, degular evolution, right? Because we're constantly evolving. I mean,
Yes.
humans used to be a lot shorter as a whole. I mean, I'm a very short person, so I clearly haven't caught up with the rest of the population. But everything's constantly in a state of evolution, right? Like you can't just say, oh, well, it's, how do you know that it's from climate change? But it's more the rate of change, the speed in which that this occurs, because the rate of change in that beach daisy was over 100 years, not that many generations. When you compare, you know, actual evolutionary timelines, you're talking millions and millions, like hundreds of millions of years. So that's my tangent.
Good tangent.
I loved it. I have a kind of quick general background question, and then we have to shift into our rapid fire round.
Okay, I'm excited.
So this is also somewhat of a tangent. We were speaking earlier about like non-traditional paths in academia,
Yeah.
and I wanted to ask you the experience of going to a school where you had hands-on experience with animals.
Ah,
How, because I'm a big fan of project-based learning,
yeah,
how much of an impact do you think that had
yeah.
on sending you down the path of ecology?
Yeah, that's a really good question. And you know what? I think it really did have a big part to play. I realized I loved getting outside. So, for backstory, I went to a high school that had an agricultural plot. Um, and I would take care of the chickens, the rabbits, the guinea pigs, help out. Um, People would make fun of me, but I did not care. So what? Screw you. I don't care. I'm going to do what I want. Um, but yeah, I think it immensely impacted how I've gone on this path of wanting to work out in nature, wanting to understand how plants and animals interact with each other, and just simply being outside. Yes, okay, as a scientist, a lot of your time, like maybe 80 to 85%, at least of my time, is spent indoors. But, you know, that 15% where you're outdoors, just it resets everything for you. And I feel like it's where my creativity really flourishes. So I really think that was really helpful for me. And
Nice.
yeah, but and I also something that I've also been saying to my friends recently is that, you know, it's sad that a lot of people feel scared to go into nature as well, because they might not feel I mean, like, they might feel like it's scary, and I totally get that. But even just going through for a walk in your local park, like,
Mm.
you know, it doesn't have to be this like far out remote regional national park with no cellular reception. Like just getting out, exploring, the coast, just being outside and getting to know nature. And then you start getting really good at observing things and spotting things. Like my husband now, he'll be, we'll be on our balcony and he'll be like, he wouldn't even see, but he can hear the yellow-tailed black cockatoos and be like, do you hear them? And I'll be like, great spot. And like, you don't even have to see things anymore to know that they're there. So yeah, I don't know. I'm just very
Absolutely.
big on people going outside, but I understand that also it
I'm
can
going
be
camping
scary.
this weekend. Absolutely agree.
Where are you going camping?
We are snow camping up in Lake Tahoe on some
Oh
place
my
called
gosh.
Iron Mountain. It sounds very intimidating, but it's just a cool name. But
I'm
yeah,
gonna Google that later. That sounds amazing.
my son is gonna go and he's digging a quinzhee. So you dig a hole down into the snow, and he sleeps
Okay.
in that. Psycho.
That is
And
awesome.
I'll be sleeping in a tent, still cold, but above
That
ground.
is so cool. And like,
Mhm.
what a great experience for your kids as well.
Mhm.
That's amazing.
I don't like being
It
cold.
feels
I
so
don't like being cold at
un-Australian,
all.
right? I
Huh?
feel like it's so foreign to Australians.
Yeah, camping in the snow. We're like, huh?
I've never done it before. I'm not looking forward to it. I'm looking forward to it. Not looking forward to it. The weather's going to be clear sky, so at night it's going to be amazing. But apart from
Oh,
that,
That
Very
sounds
nice.
incredible.
Cold. But yes,
So nice.
getting outside is incredible. It's absolutely incredible.
10 out of 10. All right. Rapid fire round. We have 11 questions and quibbles.
Okay. I don't know if you can tell, but I will rant. So, like, what's the time? You're going to have to be like, okay, done, move on.
Okay, we have 8 minutes for 11 questions because I have another meeting afterwards.
Okay. I'll be quick.
All right, rapid fire. First
Top of
thing.
the head. Go.
Yeah. What's the weirdest thing that you believed as a kid?
Oh, that people, that when you're born, you come out of your mum's belly button.
Ooh, feels very around the twist. I love it.
Yeah.
Okay. All right. That's a good one. Which teacher had the biggest positive impact on you?
My high school science teacher, Mr. Garbett, and I'll keep this quick. When I finished my PhD, I sent him an email sending him my thesis, and I was like, thank you so much. You're a legend. Feel free to read this or not. And he sent me a lovely reply. Yeah.
Oh, I love that. Shout out, Mr. Carpet.
Yeah.
All right. What is one scientific discovery that you wish you could have witnessed in person?
Ooh. Oh, that's a really good one. Maybe maybe nuclear fusion.
Ooh.
I don't know. Yeah, but you can't really see that. But maybe if I could visualize it, I don't know.
Even being in the room, yeah.
Yeah.
If you had to pick one historical scientist to go bowling with, who would it be?
Oh, that's a good one. Maybe Rosalind Franklin,
Ooh, nice.
because then we could talk about women in science and being undervalued. Sorry to take it there.
No.
She would. Oh my god, she would be so happy. Like seeing where, like, obviously we're still far away from perfection, but
we've gotten a lot better.
Yes.
Yeah, I'd like to tell her how much better it's gotten.
Nice.
Yeah.
All right, more serious questions now. If you were a sandwich, what kind of sandwich would you be?
This is you're asking the really hard ones.
Mm-hmm.
That's right.
It's tough. Nope.
Okay, when I went to Japan 2 years ago, I had this amazing egg sandwich. I don't even know what else was in it, but there was egg and something in it. So I don't know if that's a weird answer, but that's my answer.
That's a fine and unique and beautiful answer.
Thank you.
Is this the famous 7-Eleven egg salad sandwich?
No.
Seriously?
No, it's not the famous one.
Apparently in Japan, the 7-Elevens have like the best food.
7-Eleven in Japan does have the best food, and I vouch for that. But this sandwich I'm thinking of was not from 7-Eleven. Okay. It was from like some random diner. Yeah.
Wow. I've heard that the Kentucky Fried Chicken is next level in Japan. Like it's a big, big thing. But I have not heard about the 7-Eleven food.
Yeah.
Okay. More reason to go. What is your most controversial opinion about pizza?
Okay. People go to Italy and say, oh, the pizza in Italy is bad. It's boring. It's too simple. I like that. The pizza in Italy is simple and you just have like a few toppings. It's not crazy over the top. I don't know. Is that controversial?
No, that's fantastic.
It's a great answer.
It's a good answer.
Okay.
There's no wrong answers, by the way.
No. Well, I mean, some people think that pineapple doesn't go on pizza, and that's a wrong answer. But yeah.
Look, I don't know. I don't want to get into that.
We'll save that for a follow-up episode.
Yeah, that's right.
We have more rounds of these questions, so yes, if you come back, you're gonna get some good ones.
All right, what is your favorite scientific word or phrase?
Okay. I've studied like leaf defenses, and leaf hairs, like leaves being hairy, is considered a leaf defense. And some people describe it as leaf pubescence. And so I think that's really funny.
Great.
That's my new favorite one.
Pubescent leaves. Sorry, that's really gross.
That's okay. I'm going to be like stuck. 2 years from now, I'll be stuck trying to remember my PIN code in ATM, and that little factoid is going to pop into my head. That's the only thing that my brain will be offering at that time.
I'm glad.
The first person we asked these questions to was Graham Lau, and his answers were horrific. So you're remember all about ducks' special ability.
Oh, yeah.
Anyway, what is your least favorite scientific word or phrase?
I just don't like when people say put utilizing in everything. Just say used. I don't know. I don't know if that's scientific. That's not really scientific.
No, that's
I'm like,
fair.
when I
That's
read
fair.
methods and they say we utilize this, I'm like, just say we used this. Why are we utilizing? I don't know.
Yeah.
And again, don't go through my papers because if I've written utilized, probably
We'll come
one of
to
my
you.
authors
Yeah.
that put it there.
All right. What's your favorite useless science fact?
Useless. I feel like they're all kind of useful.
Good answer.
Okay.
Fair.
I don't know.
It's all useless.
Maybe that snakes are still considered tetrapods, which are four-legged animals, because they have pelvises, like
What?
a vestigial
Snakes have pelvises?
pelvis. Yeah, snakes have a vestigial pelvis, or like a leftover pelvis from when they were four-legged, but then lost their legs.
I had no idea.
I didn't know that. That's
Oh,
awesome.
really? Yeah,
Way
Google
to
it.
go.
I like it.
It doesn't look like a pelvis like you're right. It's like it's like a remnant of what a
Sure.
pelvis might be.
Sure. Cool.
Yeah.
If you could rename one planet in our solar system, what would you call it?
Do I choose which planet?
Yeah. Pick a planet.
Because
Pick
it's
a name.
not Uranus. Uranus needs to stay Uranus, obviously.
Yeah.
Maybe we change. What's a boring one? You know what? We change Jupiter because Jupiter is Jupiter named after the god of the sea? Is that Poseidon?
That's Neptune.
Neptune.
I think
Oh, so we'll change Neptune to Poseidon, the
Ooh.
original Greek. The original
Okay.
Greek.
Let's do that.
Take one away from the Romans. Nice.
That's right.
Yeah.
Okay.
Sorry to any Italians out there.
We should do that like every year, every 10 years, like switch them. Just Greek names,
Yeah.
Roman names. Greek
Yeah.
names. Just have a good time. Just have a really good time.
And then Uranus's moons are named after characters from Shakespeare. So we'll just switch it up between like Shakespeare characters and Stephanie Meyer's characters back and forth.
Yes. Oh my God. Imagine an Edward moon.
Just sparkling.
Yeah.
All right, last question. This is the big one. If you had to sing one karaoke song to save humanity from an alien
God.
invasion, all the money on this one, what would it be?
Okay. This is a controversial one because this person is not a good person, but it's R. Kelly's I Believe I Could Fly. I don't know. Am I going to get in trouble for saying that? Kovi, I just saw your shirt that you're wearing. Oh my God, he's wearing an Elas shirt, a Greek shirt. Oh my God. Is that Pythagoras on it? Yes.
It is.
Oh,
Yes.
I just noticed right at the end.
Yeah. Way to go, Cody.
It's a nice way to change the subject. But I do think that's a good song choice. Artist aside,
Yeah.
I do think it's a great song choice.
Okay, thank you.
And shout out to my mum and dad who got me this t-shirt from Rhodes.
From Rhodes?
Yeah.
Jeez Louise.
My
Papa
family
Pete.
is from an island next to Rhodes called Simi. Yeah.
No way.
Yeah.
I have an aunt named Simi. Crazy.
Do you? Oh my God. Okay, we've got to
Yeah.
continue this conversation
Absolutely.
another time, obviously.
Another time. We need at least
I just
four and
know
a
where
half
Greece
more
is
followers.
on a map. That's it. That's it. I have Greek friends, and they have delicious food.
Americans love
Gotta go.
Greece. Yeah.
I went there I went to a Greek restaurant the other week. So yeah.
We definitely have to do at least 6 or 7 follow-up episodes. This
Please.
was so much fun.
Thank you so much for having me, guys. This has been a blast. And yeah, oh my God, I could keep talking to you both for the rest of the day.
We'll definitely have you back on. Thank you for coming. For folks who want to learn more about your research or follow you on the socials, where can people find you?
So if you want to check out my research, you can look up my website, zoexiro.com. So zoexiro.com. And you can follow me on Instagram. My handle is ecologyzoe. But if you type in my full name, I'll come up on Instagram as well. So you can follow me there. I also have a TikTok that I'm really bad at consistently posting on.
Yeah, it's a pain.
Yeah.
Benjamin does the hard work of posting our content on TikTok. Benjamin, where can folks find our episodes?
Well, to carry along what we're talking about, you can find us on TikTok. And you can see us also on YouTube if you want to watch our mouths making the sounds that you're hearing.
I didn't realize I was being filmed.
Yeah.
We might just put up an image, like we can take.
It's fine.
Seriously, we're happy to put up just like a still
No,
image.
It's fine.
And you just find us Science Actually on Facebook and on
and
Instagram
Benjamin,
and everything else.
If people want to
And
find
where are
your science page, Benjamin, where can they go?
Didn't I just say it? Can you not hear me?
Oh, no, I must have had a lag. Sorry, it's the time delay to Australia.
Oh, yeah. Well, I'll say it one more time so it goes all the way around the world. You can find Science Actually on Facebook and Instagram and all the things. And now where can we find you, Coffee?
You can find me at Fun Fact Science on all the good social media platforms as well as the bad ones. Let us know if you want to hear about a particular topic or if you have a great idea for a guest for a future episode. And we will see you on the next episode of Science Actually Presents the Nerd and the Scientist.